My name is Beth Shapiro, and I am a scientist. I'm actually the chief scientist at a company called Colossal Biosciences. One of the things that Colossal is trying to do is make a mammoth, bring a mammoth back to life. But we're not doing this the way the scientists in Jurassic Park did, which would be to get a bunch of mammoth DNA sequences and shove them together, piecing together a mammoth from scratch. Instead, we start with the closest living relative of a mammoth, and that's an Asian elephant. And they already share more than 99.5% of their DNA. So we have to figure out what is that other 0.5%. What is it that makes mammoths instead of Asian elephants? To do this, we get DNA sequences from a whole bunch of mammoths from across the Arctic, and we line them up next to each other on a computer. And we compare these using a computer to a whole bunch of DNA sequences from elephants. And we look for places in the mammoth DNA where all mammoths are the same as each other, but different from their elephant cousins. And these are the parts of the genome that make a mammoth a mammoth. When we want to take a tropically adapted Asian elephant and turn it into an Arctic adapted mammoth, we have to focus on a couple of core characteristics. These are things like the woolly coat of hair that mammoths had, or thicker layers of fat that keep them warm in the winter, or other things that might have helped them be able to survive in a cold climate. For this project, we focused specifically on those traits, a woolly coat and thicker layers of fat, or different ways of metabolizing food so that the animal can stay warm. Making a mammoth is hard. One of the things that we have to do is understand how the DNA sequences that we think could change the way the animal looks actually change the way the animal looks. It would be great if we could do that experiment in an elephant, but we can't because it's an elephant, and also because elephants have a 22-month pregnancy, so it would take a really long time. A mouse, though, has a 20-day pregnancy, and after that mouse is born, it only takes another 20 days or so to see that full coat, which is hopefully woolly because of the changes that we made to its DNA. To edit our woolly mice, we took mice at several different stages. We took fertilized eggs and we took 8-cell zygotes, so a very early embryo. We used CRISPR genome editing to modify as many as 8 genes at the same time. And when these mice were born, we waited and then saw that they grew these thick, luxurious golden coats. So we know that our experiments worked because we can look at these mice and we see that they have long and luxurious golden coats. But the next step is to ask, do these woolly coats or the changes that we made to the way they metabolize fat actually affect what they can do in colder climates? And that's the next step of our research. Over the next 6 to 12 months, we'll be working with these mice, feeding them different diets, measuring how they grow, measuring how much fat they put on, and measuring how happy they are at different temperatures. So we'll find out. Our woolly mice are not a mammoth, obviously, but they are a clear step along that path. At the same time as we were working on mice, we have been doing lots of things with elephant cells in dishes growing in the lab. We've made what are called induced pluripotent stem cells, which we can coax to become different types of tissues and different types even of what we call organoids, so mini models of organs where we can test other hypotheses about how the edits we make to the DNA are going to get us closer to a mammoth. So we're working really hard on pulling together all of the edits that we need to make to change an Asian elephant into a mammoth in elephant cells in a dish in a lab. We have previously said that the first mammoth calves will be on the ground in 2028, and my team, which is the editing team, are on track to be able to deliver an elephant cell in late 2026, early 2027. But the next step is taking that cell and turning it into an actual embryo, doing embryo transfer in an elephant. And there is a lot of biology that needs to be solved there. So we're on track and we will be ready when the time comes. But whether or not everything goes to plan over 22 months of elephant pregnancy is a hard call and something that we don't really have a lot of control over. All of the de-extinction projects that we're working on at Colossal are focused on creating functional ecological proxies of species that used to be around so that we can restore the roles that those species played in their ecosystems. When we think about mammoths, we can also think about elephants. And what do elephants do in their ecosystem? They are really keystone herbivores. They're the largest thing that's out there. They're wandering around, they're churning the soil, they're distributing seeds and nutrients. When I work in the Arctic as an ancient DNA scientist, I see an ecosystem that is changing more rapidly than any other ecosystem on the planet due to global warming and climate changes that we're seeing right now. In the past, though, what we can reconstruct using ancient DNA, we see these rich mosaic landscapes with lots of different species of plants being chewed up and distributed by lots of different species of animals. And really, we don't see any of that anymore. By reintroducing these large herbivores, we will have the capacity to recreate these mosaic ecosystems. I accidentally have ended up working in de-extinction. In fact, I published a book about 10 years ago that said it was really hard and probably not a good idea. I haven't really changed my mind about whether it's hard, but I have changed my mind about whether we need these technologies. As we look across the world, we see habitats that are changing at a rate that is faster than evolution can keep up. Ideally, we could take a step back and give every habitat on the planet the time and space it needed to recover from the ways that we people have impacted these ecosystems. But we can't. What motivates me about this job, about developing these tools, is that as we get there, as we develop the ways to bring back a mammoth and a dodo and a thylacine, we will create tools that we can immediately apply to helping species not become extinct. We could, for example, change the DNA sequence of Hawaiian honey creepers to make them resistant to avian malaria, which is currently driving them extinct in their habitats. These are the same tools, gene editing tools, reproductive physiology tools, all sorts of new ideas, new ways of thinking about how we might protect biodiversity that will come to be because of the projects that are focusing on de-extinction. That is what motivates me. The idea of stopping extinction from happening, that we can make extinction a thing of the past.